Good morning and welcome to our third webinar on the value of collections webinar series, where we showcase the value of our natural science collections. We have different collections all over the country, and they are all a part of the NSCF. So the NSCF is a virtual facility that is a network of natural science collections hosts. In other words, we are a network of organizations that are holding natural science collections. And by natural science collections, we are talking about the different plants, animals, and fungi that are preserved. So basically, they are dead. They are preserved for scientific purposes, and they are also considered natural heritage as a country. So welcome, and please interact with us on our social media platforms as well as on our website. We are on Facebook, Twitter, Instagram, YouTube, and we are nscf.org.za on our website. On YouTube, you will find different videos, including previous webinars like this. So you can please go there and find the different topics that we have covered. Today, we'll be covering the iconic and historically important specimens and those that are extinct. And the presentations today will be covering those. But before we get started, I just want to introduce you to our list of guests, who this time we have quite a few. So if you can just be patient with me as I introduce them. Firstly, we have Bernard Ziffel from the Wits Evolutionary Studies Institute. And he'll be talking about the dinosaur eggs with embryos. Don't worry if you don't know what I'm talking about. You're going to understand this as he speaks. So he is Dr. Bernard Ziffel, sorry about that. And he's the University Curator of Fossil and Rock Collections at the University of the Witwatersrand in Joburg. And he curates all fossil and rock collections housed at the Evolutionary Studies Institute and the School of Geosciences. His main research interests lie in the biomechanics and evolution of the human foot, the origins of hominid bipedalism, paleopathology, and the preservation of natural history collections. Let's welcome Bernard. Moving on, we will have Fred Kikosi from the Amatole Museum, who will speak about Ubeta, the Wandering Hippo. If you don't know about it, again, he's going to explain. He is the director of the Amatole Museum, and he has 24 years experience in the museum sector. He enjoys working with people with diverse skills and backgrounds. Welcome, Fred. Next up will be Simon van Noort from Iziko Museum, who will speak about the Mbashe River Buff, extinction is forever. He was a guest in our previous webinar. If you were here, you'll remember him. Simon is a curator of entomology at the Iziko South African Museum. He holds a PhD on the systematics and phylogeny of fig wasps and he did this at Rhodes University. His research focuses on systematics and biology of wasps. He's an avid field biologist and has participated in a number of research expeditions around Africa. The discovery of and documentation of the wealth of unknown insect biodiversity or diversity is his passion. Welcome, Simon. Next up is Beth Crobola. She is from the ARC, the Agricultural Research Council. She will speak about the African Aeropoison Beetles. So her position is she's a researcher in the biosystematics division of the ARC, and she's a taxonomist who has specialized on Coleoptera, which are beetles, focusing on the Chrysomelodia, commonly known as leaf beetles, seed beetles, and longhorn beetles. She has spent her entire working career at the ARC PPRI, and it's been 35 years with the South African National Collection of Insects there at the ARC. She loves being able to work with the actual insects in the field and in the lab, and discovering new things about them, even rearing them to document their unknown life histories and host plants. Welcome, Beth. In short, she is called Beth. Welcome, Beth. And next up will be Tanya Mate. She is at the Buse Heberium. She will speak about the custodian of one of the oldest ethnobotanical plant collections. Her position is she's a curation technician, and she works on the incorporation of orphaned plant collections into the Buse Heberium. She has been working in this position for about 2.5 years, and she loves seeing and learning about all the different species. She says she especially loves to see the very old specimens, just like we'll be talking about those old specimens that are iconic today. Welcome, Tanya. Next up is Mashiane Matuhane from the Sanbi National Herbarium. He will speak about the pioneer specimens gathered at Menkiskop. So his position, he is a control herbarium technician, and his work focuses on science communication, curation, plant identifications, and research in some medicinal plants. He has been working in this field for over 20 years. And what he loves most about his work is using storytelling to communicate science, arts, and history. Welcome, Mashiane. Next up will be Greg Hofmeyer from Baywald Museum. He will speak about the southern right whale skeleton. He's the curator of the marine mammal collection at Baywald, and his focus is on pinnipeds. I hope I said that right, and has done research on them for the past 29 years. He loves the fieldwork that he does, interrogating the data to answer research questions, talking to enthusiastic fellow researchers, and following new developments in the field, and also organizing and sharing the collection. Welcome, Greg. And then next up will be Spamandla Mukeleli from SIAB. He will speak about a fish of a lifetime. He's going to unpack this so that we understand. His position is he is the curation technician at SIAB, and they are working on incorporating the Albino Museum Word collection into SIAB. Into the SIAB National Fish Collection. This is all done through processing, preserving specimen, and editing the missing gaps on the specified database. He has been hosted by SIAB for 2 years as an NRF DSI intern, and for 6 months as an NSCF curation technician. Welcome, Spamandla. We are so glad to have you. I just need to go to our last guest, who is Lemi. I seem to have lost his bio. I'm so sorry about that, but I will I will officially welcome him at the when he speaks. So we can get started with the webinar because I'll take time. Let's move to the next slide. So before we get started, we just need some webinar guidelines for engagement. So we would like you to please be kind to one another and be kind to us as the hosts, and keep your contributions helpful and considerate. You can use the chat box to say hello and let us know where you're coming from. So your organization, your school, your university, wherever you're coming from. You can do this right now as I speak. So let's use that chat box and greet one another. I think if it's not a worldly thing, but it's an African thing to get to a place and greet. So let's do that. Let's greet one another. For example, you can just say your name and your organization. So I'm Furu, from the NSCF Hub team. I see Karen has gotten us started. So thank you, Karen. And yeah, let the others join you. You can also use the Q&A box to add your questions during the webinar. I'm sure that as we go through the presentations, you're going to have questions about them. Instead of just putting them on your notebook or on your mind, please use the Q&A box to place them there. We'll make sure that they get answered at the end of the webinar. If we don't get to them, please be patient with us. You can go and place them on the webinar link, which will be on YouTube, also on Facebook, because we are live streaming to Facebook. So you can use those two platforms. If we run out of time, remember to visit our website, nscf.org.za, pardon me for that, nscf.org.za, and you'll find information regarding to natural science collections. Right, thank you for greeting each other and greeting us. I see Jermaine there. I see Faime, I see Mary, Lazarus, Liesel, Sfiso. Thank you so much, everyone. Now, if you are not aware, these webinars are connected to the showcase that we publish. So we publish it every month, hence we have the webinars every month. These showcases are on the value of collections, and we already have 3. We started with the first one on food security and agriculture, the second one on the fight against pests and diseases, and this webinar is connected to the showcase number 3, which is iconic, extinct, and historically important specimens. You can find all these showcases on the NSCF website. If you go to nscf.org.za, you go under resources, you will find the page, the value of natural science collections. You scroll down and you will find them. Now we can get started with the actual webinar, the content, the exciting stuff. Let's now welcome Bernard Ziefel. I don't know why I keep starting with a V. Forgive me for that. Bernard Ziefel from the ESI, VETS ESI. He'll speak to us about the dinosaur eggs with embryos. Bernard?
Hi, thank you, Fulu. Hi, good morning, everybody. I'm speaking on something I actually have no expertise on. I was just instructed to speak about it. So it is a bit of fun. And I happen to curate these very rare dinosaur eggs that come from Roydry. They are really quite iconic and very important, and I'm going to quickly tell you a little bit about them. These eggs come from a genus and species called Mesospondylus coronatus. They're 195 million years old, in other words, from the early Jurassic. and they come from South Africa's most representative dinosaur. We are very famous for our therapsids, our mammal-like reptiles, not so much for our dinosaurs, because they're perhaps a little less glamorous than the ones we see on TV in Jurassic Park, which is in fact Cretaceous Park. But they are probably more important because they're more basal and slightly older. So on the left here, you see a clutch of 5 eggs. They've been partially prepared out over here. And there you can see the one, which has a little skull where you can see the bottom jaw has slipped forward, and there are no teeth in the jaw, which means it looks like that particular baby needed parental care. These are also the oldest eggs in the world, not just the oldest dinosaur eggs, the oldest eggs in the world that we know of. We are sure many of the other therapsids and other taxa that are older also produced eggs, but they don't preserve. So these were found in Roydry, that is in the Golden Gate Highlands National Park. And there's a road cutting that you can see where they came from. These dinosaurs traveled across sort of floodplains, on the edges of floodplains, across desert dunes, and eventually came to this Golden Gate area to lay the eggs. So what happened was this mother laid these eggs, and as they were starting to hatch, there were 2 that kind of hatched. A sand dune came and tragically killed all of them. So none of these eggs survived, but they were actually busy. 2 of them were already crawling out of the egg. What we're fortunate is that we have an entire ontogenetic sequence of these dinosaurs. You can see the little egg on the left. I've tried to scale it to this little baby. These eggs are about 3 times the size of a chicken egg, but they turn into a substantial dinosaur, not huge, but quite big. This is the complete skeleton of an adult, and that is a reconstruction of this particular dinosaur. That is a little baby you've got over here, and that's the skull of what we call Big Mama. And to finish off, you can see over here the difference between the adult skull of a Massospondylus and the baby. And one of these very interesting things is that you've got an egg that's, you know, not that much bigger than a chicken egg, but turning into quite a substantial creature. So the size of eggs don't seem to tell us much about how big these creatures are going to get. A lot has been learned subsequently. They were taken to Canada, that was around 2000. And were prepared out there, and they've subsequently also been extensively CT scanned, with some recent research coming out done by one of our postdocs, Kimberly Chappell, with some really interesting information on the growth of these creatures. And if you do Google it, you'll be able to find out much more about Massospondylus carnatus, we having the oldest eggs in the world. If you ever come to the ASI, please come have a look. Thank you very much.
The oldest eggs in the world. Wow. You can see that we do have a natural heritage with us, a very rich one. So let's go next to Fred Kikosi from Amatole Museum, who will speak to us about Hibetta, the Wandering Hippo. I was very interested about the story in the showcase. I can't wait to hear more about it. Fred?
Yeah, many thanks, Fulu, and good morning to everybody. Hopefully, I'm not caught up in the load shedding. We expect it anytime. Hibetta, the world famous hippo. You know, why? The question, of course, I should answer first is, who is Hibetta? Hibetta, as you may see in the left-hand image, is a mounted specimen of a hippo, which occupies a special pride in our exhibitions. And it is also prominently displayed, those of you who know Amatole Museum very well, displayed right in the front of the old museum building. When you look at the logo up, our logo for more than 70 years was displaying Hibetta, but we changed it to try and incorporate the cultural history element. But Hibetta remains part of our new logo. When you look at the central part with the protruded eyes. So Hubata is very prominent. It's a draw card in as far as our operations is concerned. Then the question is why? Why is this hippo very significant in Amatole Museum's operations? There is a very interesting and fascinating story which is attached to Hubata. The story goes that around 1928, Hubata set off on a 3-year journey, moving along the KwaZulu-Natal coast, going through the former Transkei and ending up in the Eastern Cape. And it made a distance of about 1,000 kilometers. And during that time, of course, the movement was, it captivated a number of people, both locally and overseas. At some point, it was royal game. At another point, it was declared the South African national pet. And at some point, it was also declared the Union's most famous tourist. So it made a lot of headline news in a number of papers, both locally and internationally. Initially, it was dubbed Billy. And then at some point, it was called Herbert. But finally, it adopted the name Hubata when it was discovered that the hippo was a female hippo. That was only discovered at the time of her death. So this movement was very fascinating in the sense that it was attracting the communities through which the hippo was traversing. And in the process, actually, a number of local communities formulated a number of superstitious beliefs. And we are told, our literature says, there were some beliefs which were held by the Indian sugarcane workers, some superstitious beliefs held by the Zulu, the Pondo, and the Xhosa subsistence agriculturists. The hippo went as far as the Eastern Cape, but it went through a number of water bodies. At some point, it was in the Umhlanga Lagoon. At another point, it appeared in so many South Coast resorts. At some point, it also was seen in the Nahoon River, which is not far from here, which is around East London. But eventually, that marathon journey was brought to an end on the 23rd of April 1931, to the dismay of the very many communities through which Hubata had gone. So on that morning, it was found floating in the Keskama River. Keskama River around midriff is between, this was between King William Sound and Pedi, a distance of about 54 kilometers. So the director of the museum then, the director of Amatole Museum, then it was not called Amatole Museum, but the director of the museum, Captain Guy Shortridge, who was a natural historian, immediately dispatched his team to the site to try and save the skin, because of course the skin would start deteriorating. So he went there, they skinned with the help of the farmers around the area. The skin was saved and the skull and were brought back to the museum. And it is narrated that at the time when it was brought back to the museum to try and do the final cleaning, so many people were crowding the gardens of the museum to see what was going on. And during that time, because of course it was a very sad moment, because people had really been attached to Hubata. Sympathy cards were given to the museum. So many wreaths were sent as far from as Bloemfontein, and many people expressed sadness at the demise of Hubata. It is also said that the National Parliament at that stage in Cape Town, questions were raised as to what exactly had happened. which invigorated the speediness of the police to try and investigate the killing. And at the end, four farmers were convicted of the killing of Hubata. Firstly, because Hubata was protected game, and secondly, of course, these people, it is said they did not have a permit. They were fined. Of course, it may appear very conservative penalty, but the culprits were penalized 25 pounds each. Of course, that's around 1932. So certainly it was much money, but when you look at it in today's terms, it may not sound so much. Now, during the court case, it is said that so many people crowded the court, about 200 people crowded the court to see exactly what was happening. Now, of course, after Hubata had died, another debate came up. And the debate was around where should the final resting place of Hubata be. The museums and the institutions in KZN were laying claim because Hubata originated from there. The Bloemfontein Zoo also laid claim because at some point, just before Hubata was killed, they had been given a permit to try and capture Hubata and take Hubata to the zoo. And of course, Amatole Museum also placed in a claim on the grounds that they quickly responded and were able to save the skin and the skull. And of course, the museum won, and that's why we have Hubata displayed prominently in one of the galleries of Amatole Museum. But of course, not to take the whole cake away from these other institutions which were laying claim. When Hubata, the mounted body was returned from London, because the mounting, the skin and the skull were taken to London. That's where the mounting was done before the mounted body was shipped back to the Republic. So when the mounted specimen was sent back, it exhibited in Durban Museum, it was exhibited at East London Museum, it was exhibited at the East Rand Show, and it is said, it is narrated that during these exhibitions, so many people visited the institutions to see Hubata. So Hubata, as I said at the beginning, is a draw card, occupies a very special place in the museum, as well as in King Williamstown. A number of books have been written about Hubata. Some of them actually lend a kind of a cultural history component. Others lean towards the natural history. And I think the story has also done well in terms of blending these two disciplines of the museum, culture and natural history. So many books have been written, and we've actually had to, in my time here at the museum, I've witnessed about 2 book launches specifically on Hubata. The very last was a very interesting story. It was a story of the grandchildren of one of the men who had been convicted. So the story was showing how the families had been impacted by the court ruling, and of course where they are right now. There is a film in the pipeline focusing on Hubata. We have got a guest house in King Williamstown named Hubata. There are restaurants. Our newsletter is Imvubu. Imvubu in Xhosa is a hippo. So it's also looking at Hubata or focusing on Hubata. And as you saw in the beginning, our museum logo focuses and prominently displays the hippo. And our museum shop, curio shop, is also called Hubata's Hookie. So thank you so much. You are very welcome to visit us and see Hubata. Thank you very much.
Thank you, Fred. Wow. So a hippo that stole people's heart. I think that also deserves to be a title. Thank you, Fred. Now we'll move on to Simon van Noort from Iziko to speak to us about the Mbashi River buff.
Thanks, Fulu, and good morning to all the listeners and viewers. And this is one of South Africa's extinct butterfly species, and the common name is the Mbashi River buff. The derivation of this name stems from the fact that it's only known from a single locality on the Mbashi River in the Eastern Cape province of South Africa, and nowhere else in the rest of the world. The buff name comes from the fact that the species in this genus, which is the genus Delonura, there are another 6 species besides this extinct species in Africa. The genus is only found in Africa, so all 6 species that are currently still in existence are only present in Africa. And they all got this sort of pale yellow color. The other species of most of them have got actually strong black borders on the edges of the wings. So this, the Mbashi River buff is quite a plain looking species compared to the other 6 species. So this species was collected as long ago as 1863 on the Mbashi River in the Eastern Cape. And it's only known from 3 specimens collected by James Barker in December of 1863. It was described by Roland Twyman 5 years later, who was the father of lepidopterology in South Africa, which lepidopterology is the study of butterflies and moths. And Roland Twyman was a taxonomist, so he described many of South Africa's butterfly species. And on the left here is one of the original plates from the scientific publication wherein he described Delonura maculata, which is illustrated and depicted in figure 4 in this plate. And Roland Twyman actually became director of the Iziko South African Museum in Cape Town at a later period. So 2 of the 9 specimens, of male and a female, are present in the collection of the Iziko South African Museum in Cape Town. The 3rd specimen, a male, was kept by Roland Twyman in his private collection. And eventually, when he passed away, it was bequeathed along with the other specimens in his private collection to the Natural History Museum in London. So it's been almost 160 years since this species was last seen or collected. And despite numerous efforts over the last 100+ years to rediscover the species, this has been completely unsuccessful. And so the species is regarded as extinct at the moment. This is a female specimen that is present, that we have in our collection in the Iziko South African Museum. And at the time, Roland Twyman bemoaned the fact that there were so few specimens for him to base his new description on. It is definitely a very cryptic species, as the other species in the genus are as well. The image here shows the male specimen in the Natural History Museum in London. But these species are very sedentary. They spend most of their time sitting on the trunks of trees and forests and thick woodlands, and really only start flying if they are disturbed. And when they do fly, they are flying very slowly and gently around the tops of the trees in these forested areas. So there is a very, very small possibility that there is still a population of this species existing somewhere along the thickly wooded river valleys in the Eastern Cape province. So we hold out hope that we may still discover a population, but given the fact that it's been so long since specimens were seen, it's highly likely that the species is actually extinct. Thank you very much for your attention.
Thank you, Simon. Do you guys now see why it's important to have these collections? Like a species that was last seen 160 years ago. Come on. Thank you, Simon, for that. Now we'll move on to Beth Crobola from ARC to speak about the African Aeropoison Beetles. Beth?
Thank you, Fulu, and welcome to everybody online and wherever you might be. I'm going to be chatting about African Aeropoison Beetles, which people might think this is a topic that is very well known and therefore everything is known about it. So these beetles were described around about the first genus in 1833 and the second genus in 1855. So you would think that everything is known about them, but in fact, we are still discovering things. And as I take you through the talk, I will be complementing my slides with images from the Game Pass shelter in the Kamberg Nature Reserve, which I was lucky enough to visit. Diamphidia is the one genus, and I was fortunate enough to actually collect two of the species which are actually referred to as the actual Aeropoison beetles. As you will see, there are 17 described species in the world, and you will see in the map on the right-hand side, the blue shaded area is where they are actually distributed in the Afrotropical region. Then the second genus I will be talking about is Polyclada, and you will see that there are actually 16 described species. And on the map, you'll see that they occur in more or less the same area, but they also extend into West Africa, and then they are also found on the Arabian Peninsula. And I'll show you here with the cursor in Saudi Arabia and also in Yemen. Now, you will wonder why I'm not showing you museum specimens. Not because I don't want to, but because all our specimens are away on loan, and we actually have two people that are working on them. One person in Italy, actually two people in Italy, Dr. Maurizio Biondi and his assistant Paola De Alessandro. They are the specialists on African flea beetles. And then I also have Caroline Shabu, with whom I did field work, and we actually interacted from as early as 2006 when we tried to discover how far south these beetles actually occur in KwaZulu-Natal. So you will be seeing live specimens, but this is very important because we're actually trying to discover what the life histories are, what the larvae look like, what the host plants of these species actually are. Now, it would be wrong of me not to tell you about a third species, or actually a genus, which belongs to the ground beetles, and this is Lebestina. These larvae are actually ectoparasites of the larvae of the Bushman Aeropoison beetles. And although I don't know much about this genus, many people do believe that these larvae are actually more poisonous than or toxic than what the larvae are of the actual Aeropoison beetles. The two beetles I was lucky enough to work on from the genus Diamphidia, you will see here the adult beetle that I collected on Marula, and the adults will deposit the eggs on the twigs of the host branch of the host plant, and then they will cover them with feces, and these will then later harden and dry, and this protects them, they believe, from desiccation, from predators, from parasites. When they hatch, the larvae then disperse, and you actually find that Diamphidia will live as single larvae, leading a single lifestyle. But you will notice that all these larvae are covered in a fecal coat. And once again, like the eggs, this protects them from desiccation, predators, parasites, and possibly also from desiccation. So the second species that I will come up now, that I looked at, will confirm this, this life cycle. And although you'll see that the beetle is a very different looking beetle, the larva is also covered in this fecal coat, and the mature larvae will also shed the fecal coat. They're also bright red. And here you can see this larva lying on the soil as they migrate down to the soil when they actually become mature. And it's lying next to a fruit from the actual glossy-leaved porkwood. So I believe this is also partially protection, that they are actually mimicking the fruit of the host plant. Then the genus Polyclada, I collected Polyclada bahemani also on Marula, actually on the same plant that I collected the red beetle that I first spoke about. Unfortunately, I wasn't lucky enough to be able to get eggs to hatch or to actually follow the life cycle of this beetle. But I did manage to collect Polyclada flexuosa, and here you can see that the eggs are also deposited on the twigs of the host plant, covered in excrement, and that then hardens, which offers the same protection as one gets for the other groups that I've mentioned. The difference here is that the larvae actually aggregate. They live together. They are not covered by a fecal coat, but that they actually have these fecal strands which they carry with them through their life cycle. And you'll see in the last photograph that even the mature larvae carry some of these fecal strands. Now, the same as in Diamphidia, Polyclada will also migrate down the host plant stem, and then they will dig into the soil where they actually form a soil cocoon, and that they will then possibly lie dormant there. And this can last for anything from 1 to 3 years before the actual mature adult comes out. Okay, so I'd like to move on to the actual sand people. And here I've got a photograph that was taken in Botswana itself. And here you have a sand hunter who's actually digging out these pupae that can occur anywhere from 0.5 meter to 3 meters down. And they are usually in the vicinity of the host plant or below the host plant. The larvae then are actually squeezed by the bushman, and this, what we call the hemolymph, or the blood, is then actually applied to the shaft of the arrow tip. Now, what I would also like to say about the sand, and this is partially why these are so iconic and historically important. If you look at the sand hunter there, he's already wearing Western clothes. So the traditions are starting to die out, and these need to be documented. So my colleague Caroline Shabu has been working with various other colleagues. They've actually interviewed the San in Namibia and in Botswana, and there are 2 very nice papers that have come out on the traditions of the San people. And I believe this is incredibly important, because we might actually be getting to the point where we lose this knowledge. And to finish off my talk, I'd like to share with you a couple of the images of the actual Bushman hunters, or the San people that actually did the hunting. And these I took in the Game Pass shelter in the Kambach Nature Reserve. Just briefly to point them out to you, beautifully depicted. But once again, one must remember that these paintings are starting to disintegrate, degenerate due to various reasons, sometimes even human intervention. Fortunately, the University of the Witwatersrand is also very active in documenting this art, in protecting these sites. And you'll see this is, the Game Pass shelter is actually in the Maloti Drakensberg, and it has been declared part of a UNESCO World Heritage Site. So I think we are doing what we should be doing, and let's continue. Let's see how many species will remain after the revisions of these genera are done. And thank you for your attention. Many thanks.
It's so good to see the connection between indigenous knowledge systems and the collections. So thank you, Beth. I believe we're going to also see more of this in the next showcase and webinar, which will be focusing on indigenous knowledge systems. Moving right on, before we do that, I would like to apologize. We're running quite out of time, so please be patient with us. If you can just please listen in until the end. We still have quite a few people to present. I'd like to also ask them to please be a bit quick, so we don't waste people's time. Next up is Tanya Mate from Heberium to speak about the custodian of one of the oldest ethnobotanical plant collections. Tanya?
Hi everyone, thank you very much, Fulo. Heberium is custodian of one of the oldest ethnobotanical plant collections. These specimens were collected and donated by Father Franz Meyers in the early 1900s. Father Franz Meyers was an Australian missionary in the Catholic Church. He spent a lot of time in Pietermaritzburg, KZN, and he was very much interested in learning different cultures, which is why he spent a lot of time with the Zulu people, learning their language and also collecting ethnological objects, which also include this collection. The collection consists of 245 specimens, and they are all over 100 years old. They have detailed information on their labels stating the Zulu name and a description of the plant's traditional use. So here I've selected 3 specimens from the collection. The first one is the Siphus mucronata, which is also known as buffalo thorn, and in isiZulu it is called umlahlankosi. This plant has many traditional uses, but also mostly it also has many medicinal uses, but mostly in the Zulu culture, it is the branch of this plant is being used to carry the spirits of the deceased family member from their place of death to their burial sites. You may have seen this in a hospital where a family is holding a branch and leaving with it. So this represents them collecting the spirits and taking it back home. The second plant here is Atemesa Afra. This plant actually made headlines during the peak of COVID because of its medicinal uses. Also, Madagascar was also claiming that it had medicinal properties against COVID. So this is because of the medicinal uses, because it is used for colds and influenza. Because of this specimen here, we do also see that the Zulu people actually used this plant for a similar purpose. And it is also called umhlonyana in isiZulu, and in English it is called wormwood. The third one is Asparagus africanus. The specimen is used for nausea and colic, and its common name is hakdoring, and in isiZulu it is called isigobo. Okay, the value states and use of these specimens. These specimens have been well kept and can be used to verify the identities of plants of the same species collected in recent years. They are good for reference purposes. Having these specimens has actually helped in the preservation of the history of the use and the dependence the Zulu people had on plants. I believe that if we look back in the early 1900s, a lot of Zulu people were not literate, and they did not have means of keeping records except by word of mouth. So, and a lot of information in that way can actually be lost, and this can result in something like a broken telephone where information is distorted. So, an example is with my family. My grandfather was a great traditional healer. But when he passed on, he did leave some information with my grandmother, which she slightly practiced, but she was not as he was. And my grandmother tries to share some of this information with us, but she really doesn't know as much as he did, and she doesn't know how some of the plants look like. So having such specimens and such information is actually good to preserve our indigenous knowledge. And this collection has been included in the 500-year archives, which is a great platform to explore African history and culture. This is an initiative and project by the NRF chair in the Archive and Public Culture, based on the History Department at the University of Cape Town. I would encourage you to go and look at this website. There are so many things that you will see that represents our cultures, not only plants and animals, a lot of things, even clothes, what people used to wear back then. Thank you very much.
Thank you, Tanya. That's so interesting. We are back to indigenous knowledge, so we can see how the two connect so perfectly. Moving on, we're going to Mashiane Matukhwane from Sanbi to speak about the pioneer specimens gathered at Minkiskop. Mashiane?
Morning, colleagues, listeners and the watchers across the virtual land and the Facebook land. The telltale story of the iconic national Heberium, as we know it today, is never complete without talking about its origins, the people who are associated with this, and the journey it travels till this day. Now, in this short presentation, I'm going to talk about the pioneer species that we gathered at Minkiskop. For those who do not know where Minkiskop is, the picture on this slide, you can see taken from Google Maps. It actually shows you where the Minkiskop is. Currently, Minkiskop, you cannot access it like the time when the specimens that I'm going to talk about were collected. It's part of the Union Building compound, which is the national key point. The national Heberium in itself, it owes its foundings to this man called Joseph Bardavi, who was actually employed as an agronomist in the Transvaal Department of Agriculture in 1903. His interest in plants was on the economically important plants. Our records tells us that he assumed duty on the 1st of May, 1903. Something that is quite very interesting and amazing is that this man was so passionate about what he does that even in the second day in office, he went around Pretoria collecting plants. In this first expedition, one-man expedition, which he actually went into Minkiskop, we are told that 10 plant gatherings were made. And I here to tell you that from these 10 plant gatherings, this is how the origin of the national Heberium started. By the time Bardavi actually started the Heberium, the building that you see on the top right corner is called Folkstem Building on the Pretoria Street, right in the city center of Pretoria. This is exactly where he was based until his retirement in 1913. Now below here, you will see the 4 specimens in display. These are part of the 10 gut plants that he collected on that first expedition. Now, his name or his contribution is actually honored in the naming of the 3 species, the Aludaviana, Bachelia davi, and the Ficus pardavi. Now, if you look at the journey from Folkstem building and up to today, where we are today at the Silverton Hill, I would like to tell you that there are so many stories that we can tell about this journey. However, let me choose few of them. When the Heberium started, it wasn't called National Heberium. It was just called Heberium. However, in 1905, it was registered as a colonial Heberium. That's when it was still at the Mingis, at the Folkstem building. However, as the collection grew, as you have seen earlier on, I said that he collected over 14,000 specimens. The collection was actually becoming quite huge and the building was very small to actually carry that. Then they moved to a building called Freire Hayes, which is on the western side, right at the Union Building grounds on the western side. That's where the Heberium was. Something very interesting also happened when the Heberium was still there. In 1918 is when it was officially named the National Heberium. However, as the collection grew, become bigger and bigger, the space was also becoming a problem, and it moved in 1973 on the 2nd of July to the current location where we are today. But something quite very interesting is that when Pardavi actually retired in 1913, the man who took over from him is Dr. Paul Evans. Talking about Paul Evans, how can we not talk about John Christian Smarts? Who is John Christian Smarts? John Christian Smarts, if you go into history, it will tell you that he's a statesman, a soldier, a philosopher, and a botanist. And this man was, he served South Africa as a prime minister 2 times, in 1919 to 1924, 1939 to 1948. And he also played a leading role in the formation of what was known as the Union of South Africa in 1910. The Union of South Africa, actually, if you know South African history very well, you will know it was actually a union of the 3 provinces, the Natal, Cape, Transvaal, and the Orange Free State. Not only that, he also played a role in the formation of what was known as the League of Nations, which is the current United Nations. Back to botany. This man, he was not only a statesman, he was also a botanist. He also was one of the people who associated with the National Heberium. He was also a friend of Paul Evans. Now, in his spare time as the Prime Minister of South Africa or as a politician, he will go with Paul Evans into collecting specimens, of which at the time of his death, he already collected over 4,000 specimens, which are now housed in the National Heberium. Now, his contribution also, he is honored in the naming of the 2 species, the Titularia smartii and the Teronia smartii. Now, the 2 pictures that you see on the slide, the one is actually a book about Smarts, the botanist, who was also the Prime Minister of South Africa. Now, the image that you see at the bottom there is a specimen, an example of a specimen that he collected along the Pinarus River in Warmbad in 1932 October. Now, when looking at this story of how this Heberium started, it brings to mind the sayings of the Chinese when they say a journey of 1,000 miles begins with a single step. I would like to say to you that the Heberium, as you see it today, it actually started with 10 specimens. Now, since its establishment, it has accumulated approximately 1.5 plant specimens, collections over more than 200 years, with its oldest specimens dating back to the early 18th century, 1800s to say. This oldest specimen is actually collected by the legendary explorer, William Bushell, He collected this on the 18th, on the 31st of January, 1811. And now the National Herbarium, from that 10 gatherings, it has actually grown to become what we will call it the largest herbarium in Africa and the largest fourth herbarium in Southern Hemisphere. As the National Herbarium, we are proudly the leading institution of plant diversity in Southern Africa. Thank you very much for listening. I would like to end this by saying that from this story, we can actually see that there was something about our natural history collections in time past, wherein even powerful people wanted to associate with our collections. Thank you for listening.
Thank you so much, Mashiane. What a history you just showed us. I think most of the people here didn't even know anything about it. I know I didn't, so thank you so much for that. So we'll skip Greg and go on to Spamandla Mkeleli from SIAB to speak about a fish of a lifetime. Spamandla?
Good morning, everyone. Welcome to the webinar. The NRF SIAB National Fish Collection represents the largest collection of African fishes in the continent and is the most comprehensive collection of Southern African fishes in the world. The collection comprises of over 1 million specimens that represent more than 5,000 species, with the oldest specimen dating back to the 1800s. Unique to the SIAB National Fish Collection is the only record and specimen of albino great white shark. The shark was caught off Bogness in the Eastern Cape by Els in 1996 and was donated to SIAB in 1997. The shark has an extremely rare recessive condition, meaning it lost all the pigment in the skin. Normal great white sharks have a grayish dorsal surface and a pale to white ventral surface, allowing them to camouflage. The albino shark is all white and cannot camouflage. This makes it hard for them to hunt, and yet it gets it makes it easy for them to get preyed upon by predators. The albino great white shark is an iconic record because they are scarce. Normally, great white sharks are born measuring between 109 to 165 centimeters, and our albino specimen is 158 centimeters, meaning it's a baby great white shark. The specimen has an associate tissue sample logged in the aquatic biobank of the NRF SIAB. Sharks live up to about 70 years, but that is a rare thing for the albino great white sharks. Hence, we believe our specimen represents a fish of a lifetime in our collection. Thank you.
How interesting. I never knew there was an albino shark. Thank you, Spamandla. So I see Greg is back. So we'll move on to Greg to speak about the southern right whale skeleton. Greg is from Baywell Museum. Greg?
If I can just start with this wonderful specimen here. It hangs from the ceiling in the Marine Hall at Baywell, and it's one of our most impressive exhibits. This is specimen number PMN0019. It's the 20-meter long, complete and articulated skeleton of a southern right whale. And it's only the second marine mammal specimen that was collected by the museum, arriving in 1901. At the time, there was still an open boat whaling industry in Algoa Bay. When whales were sighted in the bay, that's Algoa Bay, from the hills surrounding what was then Port Elizabeth, now the city of Gqeberha, Small boats would set off from the shore. This was a dangerous business, with the whalers using hand-launched harpoons to kill the whales, and many of them lost their lives. If they were successful, the dead whale would be towed back to shore and be dismembered by many of the various whaling firms based in the bay. On the 12th of November 1901, a whaling boat belonging to the firm Messina Brothers brought this particular whale back shore. It was pulled ashore where the 6 pillars now stand, and the skeleton was cleaned off before being transferred to the museum. This skeleton has traveled with the museum wherever it has been and has been displayed throughout that time. This skeleton serves to illustrate while real items in museum exhibits cannot be replaced by online displays or photographs or descriptions in books. When I tell people what the skeleton represents, they are often struck by its size and are amazed that it comes from such a huge animal. Walking around it or under the animal gives them a great sense of what exists in the depths of the ocean. While we can describe whales in terms of lengths of buses or weights of elephants, it doesn't really compare to seeing the actual object. And then I use this as an introduction to the importance of the ocean, the life in the ocean, and the impact also of human beings' unbridled exploitation on marine life and the need for conservation. And also for the natural history collections, both at Bayweld and further afield. Our behind-the-scenes tours start under the whale, with students, school children, or members of the public open-mouthed, craning their necks upwards to have a look at this enormous specimen. And they are then eager to go down into the dungeons to be shown the various specimens in the collection and to hear about the work that we're doing. This specimen is also not only useful for education, but also for research. And we've been visited by other researchers, most recently a Belgian group who wanted to take photographs of the whale to try and identify. They were specifically looking for an articulated southern right whale to see how these animals move through the ocean, how their enormous shape allows them to move through the ocean. So our world has changed a lot since this enormous whale met its sad end in the bay. And there's a greater scientific understanding of the environment and our impact on it. But when I take people to view this whale, I feel that still, 120 years later, it still continues to have the same impression that it did on people when it was first collected. And it makes people wonder, and it inspires scientific curiosity in older researchers or eager young researchers, all the same. Thank you very much.
Greg, you just reminded me of how I felt the first time I was in a museum, especially ones that have those huge skeletons. So thank you for such an interesting presentation. We will move on to our last speaker, who is Lemi Mashinini from the Ditsong National Museum of Natural History. You'll remember I didn't give him an intro, so I'll do that now. So Lemi is the curator of herpetology at Ditsong. His research focus is on ecology and taxonomic review of the Agama aculeata species complex. He's been in the field for more than 10 years. And what he loves most about his work is going out for field work and coming back with pockets full of a bunch of kicking lizards. Talk about a person who's interested in herpetology. Of course, he has to be excited about lizards. Welcome, Lemi. So Lemi today is going to speak to us about the extinct Eastwood's long-tailed seps. Take it over, Lemi.
Good morning, colleagues. As Fulo said in the introduction, my name is Lemi Mashinini, curator of herpetology at the Ditsong National Museum of Natural History that is based in Pretoria. My presentation is going to be based on the extinct Eastwood's long-tailed seps, scientifically known as Tetradactylus eastwoodi. And since the discussion is based on an extinct lizard species, I think it is important to briefly look at factors or threats that result in reptiles population declines and their eventual extinctions. One of the factors that is responsible for the decline in reptiles population is pollution. Pollution can take various forms, the commonest including noise and chemical pollution. Noise pollution threatens species by altering their natural behavior. And as we all know, thousands of chemicals are discharged into the environment, and their lingering presence may threaten biodiversity, thereby affecting species and degrading their ecosystem. The second factor is human activities, especially those relating specifically to recreational activities of people that spend time in nature, traveling in vehicles of established transport corridors may alter, destroy species habitat. The third factor that I want to talk about is harvesting or over-exploitation by way of hunting, trapping, and unsustainable harvesting, which is a serious threat to some of the reptile species. Examples being pythons and crocodiles that are often hunted for their skins, and sea turtles being hunted for food by some communities. We also have accidental mortality as a factor, where reptiles are killed while crossing roads. And this often leads to local extinctions of reptiles. The other factor is climate change. As a growing extinction concern, climate change is increasingly implicated in the decline of vertebrates, including reptiles. In South Africa, factors predicted are increased temperatures and reduced rainfall, which will result in change in species' natural behavior, alter or reduce their habitat structure, thereby posing a risk to their survival. Another factor is invasive alien species. The introduction of invasive alien species is a threat to native species. Their impact range from predation on local populations, increased food competition, and also introducing diseases and pathogens which negatively affect the viability of native species. And the last factor or threat is habitat loss and degradation. This includes agricultural activities, including afforestation, extraction activities such as mining, fisheries, and timber logging, and development activities such as development of new settlement areas, industries, and agriculture. Thirdly, this factor is the one that is responsible for the extinction of Tetradactylus eastwoodi that I am presenting on today. The National Museum of Natural History curates the only two surviving specimens of Tetradactylus eastwoodi, shown by the picture in the first slide. Both specimens were collected in the woodbush granite grassland in the Limpopo province. The area where this vegetation type occurs is shown in red on the map in the top picture. And there are about 14 vegetation types in South Africa that have been recorded to date. And wood bush granite grassland is one of those. So the area that you see in red in the top picture is the one where this vegetation type occurs, and this is the area which was occupied by Tetradactylus istudi. The first specimen was collected in 1911 by Mrs. Istudi, after whom the species was named. And the second one was collected in 1928 by Mr. Vincent Weger. After the collection of the two specimens, there were several subsequent searches in the area, but all the efforts were unsuccessful as no specimens were collected or seen. Until the species was officially declared extinct by International Union for the Conservation of Nature, shortly known as IUCN. Sadly, this vegetation type, which is endemic to the area, is also considered critically endangered, which further eliminates the possibilities of the species ever being rediscovered. If you look at the bottom picture, the picture shows how the general area where the species used to occur has been altered, with original vegetation replaced by pine and blue gum trees plantations. Some of the factors attributed to the vegetation type being critically endangered are transformation through development, farming, and plantations, invasive alien vegetation, illegal grazing by domesticated grazing, human activity, which includes littering, dumping, illegal fires, driving on grasslands, etc. Effects of climate change, illegal and unsustainable harvesting of medicinal plants, and lastly, encroachment by woody plants. In this slide, which I believe is the last one, I just want to touch on the role that natural history museums and their collections, of course, play in species conservation. As we all know, the traditional function of natural history museums is to collect, curate, and preserve specimens. The specimens include those of species that are critically endangered, protected, threatened, and we also collect species that are of low conservation concern. Now, when species become extinct in nature, natural history museums become extremely important in the sense that they would often have preserved material of extinct species or specimens that will serve as reference material for various studies, such as morphological studies, spatial distributions, habitat preference, and are also able to provide material needed for DNA studies. Now, in the case of Tetradactylus istudi, thanks to the Ditsong Natural History Museum, for being able to preserve the specimens and their associated collection data. Also, thanks to the collectors of the two specimens, Ms. Istudi and Mr. Vincent Weger, who upon collecting the specimens saw the need for them to be deposited in a museum, for their data to be captured and for them to be preserved. Without the efforts of both collectors in ensuring that the specimens are deposited in the museum, and the museum for curating and preserving the specimens, the herpetological community wouldn't know that such a species once occurred. So my last statement, colleagues, will be to invite colleagues who have interest to see the specimens, for them to come to the museum and actually see the specimens. Thank you very much.
Thank you, Lemmy. I just want to reiterate what you said. If the specimens were not collected, we wouldn't have an idea of the existence of that specimen. So even though it's extinct, we still have records of it. We still know what it looked like. So just a thanks to everyone in the collections space. Thank you for the work that you do. Thank you so much to all our presenters. I just wanted to put some apologies for some of our speakers that didn't make it. So there's Luan Ketten from East London Museum who was supposed to join us, but because she fell ill, she couldn't join us. And David Allen from the Durban Natural Science Museum. He was supposed to present, but he couldn't make it. About David, also just a note that he has just recently retired. So I just thought it was important for us to just, I don't know what we would call it, but I don't want to say goodbye to him because I don't want to lose him. I think many of us understand he has done great work in the field and in this community, and he was contributing so much to the NSCF with his articles, with his research, and he was also very open to engaging. Just like in this webinar, he really wanted to do it, but just circumstances didn't allow him. So David, if you watch this, thank you for all your work and we'll miss you. Now, if any of you have any questions, I see there's nothing in the Q&A box. So I don't know if that means you don't have any questions. Yeah. Thank you, Mary, for the correction. Luan wasn't sick. It's her father who passed away. So our condolences to her and her family. Andrew says David will be back. So it's good to know. It's good to know. Right. No questions. I'm just gonna let you guys give you a minute if you might have any question. No questions. Whenever there are no questions, I just consider it as the presentations having been very clear. So they explained everything so clearly to a point where you don't have anything to ask. So well done to all our presenters. Now we will close the webinar. Thank you for sticking with us, even though we were behind time. We appreciate that. Thank you for attending our webinar, and we hope you will come back next month, when we will be speaking, not next month, maybe still in October, but we'll be covering indigenous knowledge systems. So we hope you'll join us. Please do remember to interact with us on our social media platforms. We are NSCFSA on Facebook, NSCF_SA on Twitter, NSCF_SA on Instagram, Natural Science Collections Facility on YouTube, and nscf.org.za on our website. So we hope we'll see you there. Thank you so much and have yourselves a great weekend. Thank you.